Hello friends, welcome back. So now the break is over and let's start. We were talking about the lipoprotein before the break. So we had mentioned very briefly what exactly is a lipoprotein, what function it is performing very broadly. We looked at the structure of the lipoprotein, the outer and inner layer components, and we briefly mentioned which are the important lipoprotein that you should know about. Now, if you look at the nomenclature of the lipoprotein, if you look at these names, chylomicron, VLDL, LDL, HDL, obviously these names are based on density of the lipoprotein, and this is how in the past the lipoproteins were separated, and it was criteria for labeling the lipoprotein. So, how do we separate the lipoproteins in the past? By the technique called ultracentrifugation. The technique called ultracentrifugation, which is based on density. This ultracentrifugation is based on density, and you'll find here, if you take a tube in which you put the sample and then you carry out the ultracentrifugation, means you rotate this tube at a very high speed, so that there can be separation of the components. So what will happen, let's say this is the surface of the sample. So one of the lipoprotein layers will stay close to the top. Because it is very light, it will stay close to the top. It will not settle even after centrifugation at a very high speed. One of the layers would be very much lower down in the tube. Because of its high density, it will quickly settle to the bottom. One of the layers will be at the bottom, and we will have two layers somewhere in between. Two layers somewhere in between. This is arranged as per density. The most dense will go at the bottom, the lightest one will stay at the top. This means the layer at the bottom must be a high density lipoprotein. The layer at the top is the lightest, which is called the chylomicron. Just above the high density lipoprotein will be the low density lipoprotein, and even lighter than low density lipoprotein will be very low density lipoprotein. So in the past we used to separate the chylomicron on the basis of ultracentrifugation, which uses the property of density, and the various level layers were labeled as HDL, LDL, VLDL, and the chylomicron. But nowadays we don't use the ultracentrifugation. What we use now is electrophoresis. Now, separation electrophoresis is not dependent on a single factor. It is dependent on multiple factors. What is the charge? What is the protein content? What is the ratio between the charge and the protein content? What is the ratio between, what is the size? What is the density? So multiple factors come into play. Remember, the separation via electrophoresis is not dependent on a single factor. It depends on multiple factors. Broadly speaking, among all the different points, most important is the relative protein content. But please note, this is not the only factor which is determining the separation electrophoresis. There are other factors. What is the size, what is the charge on the proteins which are there? So various factors come into play when you are doing the electrophoresis, not just the relative protein content, but this is by far the most important consideration when we are doing the electrophoresis. So when you do the electrophoresis and carry out the separation, let's say you apply the sample here and then you carry out the separation. So separation will again give us four layers. One of the layer is very close to the initial point of application, doesn't move much. One of the layer will run off very far towards the end, and we have two more layers somewhere in between. We have two more layers somewhere in between. Alright, now here if you look at the first layer, it is same as in ultracentrifugation, that is the chylomicron. If you look at the last layer, it is the same as you see in ultracentrifugation, that is the HDL, but the intervening two layers are different. The VLDL will move faster than LDL. The VLDL will move faster than LDL. If we just look at the protein content, we will find that the VLDL has less total protein than LDL. But the nature of protein, the type of charge, the size of the molecule, all of these are factors. All of these are factors in faster movement of VLDL in the gel. Now, on electrophorese, the fragments that you get are also slightly differently labeled. This is the alpha lipoprotein. This is the pre-beta lipoprotein, and LDL is called the beta lipoprotein. Okay, this is a slightly additional, or you can say different labeling which is seen. Sometimes, depending on the content, you may find a faint line in between the pre-beta and the beta lipoprotein, that is between the LDL and VLDL, which happens to be ideal. Depending on the amount of ideal, sometimes it may appear on electrophoresis. Most frequently, it is not there, but nevertheless, when it appears, it will lie in between the two layers as shown here. Alright, so this is the current method for separation, that is the electrophoresis, and the older method for separation, that is the ultracentrifugation. The older method for separation, that is the ultracentrifugation. Alright, so ultracentrifugation and the electrophoresis. Okay, so now we have seen how we can separate the lipoproteins, traditional separation as well as the current method which is used for separation. Let's talk about some of the important points related to the lipoprotein. So what we'll do, we'll make a short table for the lipoproteins. We'll make a short table for the lipoproteins, and we'll describe various points for the lipoprotein in this table. What are those points that we are going to describe? Starting with the name, then from where is the lipoprotein synthesized? From where the lipoprotein is synthesized? What is the function of the lipoprotein? What is the function of the lipoprotein, and how do you distinguish this lipoprotein from others? In other words, what are the marker proteins, marker apolipoproteins which are there on the surface? The marker apolipoproteins which are there on the surface. So we'll draw a short table highlighting these points. Let's see. Okay. So first we start with the chylomicron. We start with the chylomicron. Chylomicrons are synthesized by the intestine. They are synthesized by intestine, and their primary role The primary role of the chylomicron is the transport of the dietary or the exogenous triacylglycerol. They are responsible for the transport of exogenous triacylglycerol. If you look at the content, you'll find the primary content is the tricycle. So obviously other components are there, but they are there in lesser amount, so they are not the prominent component. The prominent component happens to be the triacylglycerol. It is written at the top, so I'll not mention it here, just for reference. And the marker for chylomicron is the incomplete apolipoprotein, that is the B48. B48, the incomplete apolipoprotein, is the marker for chylomicron. Now, once this chylomicron delivers its triacylglycerol to the tissues, it becomes smaller in size, and it is subsequently known as the chylomicron remnant. It is subsequently known as the chylomicron remnant. So obviously it comes from the chylomicron. The source is the chylomicron. And if you look at the components, it will have similar amount of the cholesterol, sorry, and the triacylglycerol. All right, the cholesterol and triacylglycerol, and even the phospholipids, because most of the triacylglycerol has already been delivered. So the phospholipid and cholesterol, which were there in less amount, now become comparable to the triacylglycerol content. Whenever we talk about remnant, remember the primary marker will be there, but in addition to primary marker, we will always have the apolipoprotein E. E is the hallmark of the remnant lipoprotein. Alright, so whenever we talk about the remnant lipoprotein, we must have the apo E. Okay, so this is the chylomicron and the chylomicron remnant. Let's move on. Chylomicron and the chylomicron remnant. Next, we have the VLDL, very low density lipoprotein, synthesized by the liver, and it is responsible for transport of endogenous triacylglycerol. This is the triacylglycerol which is synthesized by the liver for the extravascular tissue. So it is transporting the endogenous triacylglycerol, and its marker is B100. But in addition to B100, you will also find the various types of ApoC, C1, C2, C3. They are also present. So multiple apolipoproteins on the surface of VLDL are a significant point. The VLDL will then get converted into the VLDL remnant. It gets converted to the VLDL remnant. If you recall, we said the VLDL remnant is also known as IDL. So VLDL remnant, known as IDL, obviously synthesized from the VLDL, and just like chylomicron remnant, it will have now comparable amount of cholesterol and the triacylglycerol. It will have the comparable amount of cholesterol and triacylglycerol. And a remnant, in addition to the primary apolipoprotein, will also have ApoE. It will have the ApoE. We also said that the VLDL sometimes may be converted VLDL may be converted into the LDL. So next one is LDL, and LDL is also coming from VLDL. However, the LDL is responsible for delivering the cholesterol to the extrahepatic tissue, so it is responsible for transport of cholesterol. So remember, LDL doesn't directly mean the atherosclerosis. LDL has a physiological role. What is the physiological role of LDL? That is the transport of cholesterol to the extrahepatic tissue from the liver. I will repeat, physiological role of LDL is transport of cholesterol from liver to the extrahepatic tissue. And if you talk about marker, it has a single copy of B100. A single copy of B100 is the hallmark of LDL. Alright. And last we come to the HDL. Interestingly, HDL is synthesized by both the small intestine and the liver. It is synthesized by both small intestine and liver. It is responsible for the transport of cholesterol from extrahepatic tissue to the liver. We call it the reverse cholesterol transport. Normally all the lipids are going to the extrahepatic tissue, but HDL is taking the cholesterol from extrahepatic tissue back to the liver. Now the marker for HDL is A1, but it contains all apolipoproteins except the B series. Except the B series, it will contain all the apolipoproteins. This is the hallmark of HDL. It is rich in apolipoproteins. It is rich in the apolipoproteins. So this is a very brief table that you should keep in mind when you're talking about the lipoproteins. If you remember this table, then you are able to comfortably answer most of the questions coming from the lipoproteins. Most of the questions coming from lipoproteins you can quickly correlate with this table. You can see from where they are synthesized, what is their primary function, what is their content, what are the different apolipoproteins which are there on the surface. All of this is described in this particular single small table. This single small table is able to clearly tell you all the points related to them. Now one extra point which I will mention here, these markers which are there in the list, they are actually ligand for the corresponding receptors. They are readily identified by the corresponding receptors, which helps in uptake of these lipoproteins. So I'll quickly highlight the markers. Okay, B48, I told you it is an incomplete apolipoprotein, so it doesn't have a receptor. The B48 is an incomplete apolipoprotein, so it doesn't have a receptor. ApoE is identified by two receptors. It is identified by the LDL receptor and it is identified by LRP. LRP stands for LDL receptor related protein. So it is a protein similar to the LDL receptor performing a similar function. Alright. The B100, rather I would mention the B100 here. The B100 is identified by LDL receptor. B100 is identified by the LDL receptor, whereas the ApoA1 is identified by the HDL receptor. So these are the different receptors for the corresponding markers. These markers are called ligands when they interact with the receptor. When there is interaction with the receptor, in that case we call them the ligand, otherwise we call them the marker. They help us to identify the different lipoproteins. When they are interacting with the receptors, they act as ligands, so they can also be called ligands. Remember, b48 which is there on the chylomicron, that is an incomplete protein, and therefore it doesn't have a corresponding receptor with which it can interact. Now one very important point, or you can say distinction, between B48 and B100. There is a lot of confusion among the students related to this point. Even the confusion is there in the books. Even the same textbook sometimes mention the different description for these two proteins. So I'll quickly show you. Please understand carefully. DNA. From DNA, after transcription, you get the heterogeneous nuclear RNA. The heterogeneous nuclear RNA undergoes post-transcriptional modification and it forms the messenger RNA. Right? This messenger RNA will go out from the nucleus into the cytoplasm, and in the cytoplasm it will undergo transcription. Alright, let's talk about the gene for ApoB. Let's say this is the gene for ApoB. So we get the messenger RNA for ApoB. Alright, in the liver, the transcription is complete. In other words, 100% transcription takes place and we get B100. This is occurring in the liver. In liver, the transcription is complete 100% and we get the B100. However, in small intestine, there is a phenomena called RNA editing. The result of this RNA editing is stop codon is introduced at 48% of the messenger RNA. At 48% of messenger RNA, result only 48%. I'm sorry, this is not transcription, this is translation. I'm so sorry. 48% translation occurs, because at that point there is a stop codon. When the stop codon will come, the further translation cannot take place, and the resulting protein is therefore called B48. I hope this is clear to everyone. The 148 merely denotes what percentage of the ApoB messenger RNA has been translated. It is in no way related to the mass of the protein. So the books will say B100 mass is 100 kilodalton, B48 mass is 48 kilodalton. That is not correct. Please note, that is not correct. The 148 relates to what percentage of messenger RNA is getting translated. What percentage of the messenger RNA is getting translated, from where we get the B48 and the B100. This is very, very important. A lot of books will make confusion at this point. Okay, let's come back to this table. We have seen the different lipoprotein. Now I'd also like to talk a little bit about the protein part, the apolipoprotein. We'll quickly talk about the apolipoprotein part. They have several important functions. Let's take a look at the apolipoproteins. Let's take a look at the apolipoproteins. Alright, let's talk about the functions of the apolipoprotein. Now, besides being ligands, which we already discussed, the apolipoproteins can also perform other functions. They can act as enzyme cofactors, or in other words, they can act as activators of the enzyme. They can act as activators of the enzyme. The ApoC2 is activator for lipoprotein lipase. Lipoprotein lipase is an enzyme which is attached to the endothelium of capillaries. It breaks down the tracylglycerol in the lipoproteins. So the ApoC2 is activator for lipoprotein lipase. ApoA1, it is the activator for lecithin cholesterol acyl transfer. We already seen the LCAT, what it does. We already seen how the LCAT is functioning. It transfers the fatty acid from lecithin to the cholesterol and converts it to the cholesterol ester. We already seen the function of the LCAT. The LCAT is activated by the ApoA1. ApoA1 will activate the LCAT. Then besides being activators, they can also act as inhibitors. The apolipoproteins can act as inhibitors. The ApoA2 and ApoC3 are inhibitor for lipoprotein lipase. Please note, ApoC2 is activator and C3 is inhibitor for lipoprotein lipase in addition to the ApoA2. ApoC1 on the other hand is inhibitor for cholesterol ester transfer protein. I'll show the function of cholesterol ester transfer protein. The cholesterol ester transfer protein will transfer the cholesterol ester from HDL. From HDL, it will transfer the cholesterol ester where to VLDL. It will transfer the cholesterol ester to VLDL, but obviously there is nothing like free lunches. There is nothing in freely available. So in exchange for the cholesterol ester, the VLDL will provide the triacyl glycerol to HDL. That is why when you look at the mature HDL, it should ideally have only the cholesterol and cholesterol ester, but it also has a significant amount of the triacyl glycerol. This triacylglycerol is coming from the VLDL. So this is mediated by the cholesterol ester transfer protein. This is mediated by the cholesterol ester transfer protein. Alright. So the CETP is inhibited by ApoC1. CETP is being inhibited by ApoC1. So the apolipoproteins can function as activators, they can function as inhibitors, and lastly we've already seen they are working as the ligands. They are working as the ligands for the various receptors. ApoB100 and ApoE are the ligand for LDL receptor. ApoE is also the ligand for the LRP, the LDL receptor related protein, and ApoA1 is the ligand for the HDL receptor. ApoA1 is the ligand for HDL receptor. Now that we know the functions of the different apolipoproteins, we have seen the structure of the lipoprotein, we have seen the role, the site of synthesis, the components of the lipoproteins. We can quickly discuss how the different lipoproteins are transporting the different types of lipids in the body from one place to another. Alright, let's take a look. Starting with the chylomicron, let's first talk about the transport of lipids by chylomicron. We already said the chylomicron is responsible for transporting the dietary triglycerol. Alright, you can see here the dietary triglycerol is there. It is digested in the small intestine. From small intestine, it is repackaged and released in the lymphatics. That is why we call it the chylomicron, the small particle, micron particles in the chyle. That is the chylomicron. It is responsible for the milky appearance of the lymphatic fluid after the meal. Alright, so chylomicron released in the lymphatics. This chylomicron contains the B48, which is the marker. It contains various types of ApoA, and majorly it contains the triacylglycerol. Some amount of cholesterol is there, but majorly it contains the triacylglycerol. However, this is called, you can see, the nascent chylomicron. This is a nascent chylomicron. In other words, it is the inactive chylomicron. Why inactive? Recall we had said ApoC2 is required for activating the lipoprotein lipase. As long as ApoC2 is not there, the lipoprotein will be inactive. So next step, we have to borrow the ApoC2, which will come from HDL. ApoC2 is borrowed. Along with ApoC2, ApoE is obtained, and then we get the active or the functional chylomicron. Once the ApoC2 becomes available, then we get the active or the functional chylomicron. Now at this point, see what happens. Now the active chylomicron, when it reaches the tissue, the ApoC2 will activate the lipoprotein lipase, which is attached to the walls of the capillaries, right? This activated lipoprotein lipase in turn will act on the triacylglycerol. It will act on the triacylglycerol and cleave it into two. It will cleave it into two, the glycerol and the free fatty acid, which will quickly be taken up by the tissues for their own use. So ApoC2 activates the lipoprotein lipase. Lipoprotein lipase acts on the triacylglycerol in the lipoprotein. That is why it is called the lipoprotein lipase. The lipase which is acting on the lipoprotein is the lipoprotein lipase. It cleaves the triacylglycerol into the glycerol and the free fatty acid, which will go into the extrahepatic tissue. The glycerol via circulation reaches back to the liver, where it can be reutilized. Alright, so the primary role of the chylomicron, you can see, it is taking the dietary triacylglycerol and delivering it to where? The extrahepatic tissue. It is delivering them to the extrahepatic tissue. Now once most of the triacylglycerol is delivered to the extrahepatic tissue, this spherical structure which was there, the chylomicron, the content is less, so the sphere will become smaller. When the sphere becomes smaller, the surface area will also become smaller, and it is not able to support so many proteins, B48, ApoE, ApoA, ApoC2, all of these cannot be supported. So some of the protein must be discarded. The proteins which are discarded are ApoC2, no longer required, along with ApoA. Why ApoA? Because the B48 is an incomplete ligand. It will not be recognized by receptor. So how will you uptake the chylomicron remnant? Via the ApoE. So ApoE is retained along with B48. B48 is integral. So B48 and ApoE are retained. At this point, we get the chylomicron remnant. Recall in the table we had said the chylomicron remnant has comparable amount of the triacylglycerol and the cholesterol. Comparable amount of triacylglycerol and the cholesterol. Now when this reaches the liver, we have two types of receptor. The LDL receptor, we said LDL receptor will identify the ApoB100 and ApoE. Here we have ApoE, so it will identify the chylomicron remnant. Similarly, LRP is specific for the ApoE. So LRP will identify the ApoE and take up the chylomicron remnant. Now one very important point which I want to highlight here. The LDL receptor is present on the extrahepatic tissue also. Please note, the LDL receptor is present on extrahepatic tissue also, but this LDL receptor is not able to take up. It is not able to take up the chylomicron remnant. Why? Reason being, The chylomicron remnant is a large particle, although it is much smaller than the chylomicron. Chylomicron itself is very large, so remnant is still significantly large molecule, and the membrane of the extrahepatic tissue is not flexible enough to engulf this large chylomicron remnant. Whereas when it comes to the liver, when it comes to the liver, in the hepatic sinusoids there is a lot of flexibility in the membrane, so even big particles can be engulfed in the hepatic sinusoids. That is how the chylomicron remnant is taken up by the hepatocytes, but it cannot be taken up by the extra hepatic tissue because less flexibility is there. So that's about the chylomicron. I'll repeat: dietary triacylglycerol packaged into the chylomicron and released in the lymphatics. This is the nascent chylomicron. It takes up the ApoC2 from HDL, becomes the active chylomicron, goes to the extrahepatic tissue where it delivers the triacylglycerol by the action of lipoprotein lipase, gets converted to the chylomicron remnant, which is taken up by the liver. Very simple few steps for the delivery of exogenous or the dietary triacylglycerol to the extrahepatic tissue. Something very similar also occurs for the VLDL. Very similar for VLDL. Let's see what is happening in VLDL. What are the points of difference? First point, the VLDL is synthesized. The VLDL is synthesized the VLDL is synthesized by the liver. It is not synthesized by the small intestine, it is synthesized by the liver. Alright, because it is being synthesized by the liver, it has large number of proteins. It even has ApoC but not ApoC2. C2 is not there. C1, C3 can be there, but C2 is not there. And the apolipoprotein which is synthesized is the B100. In chylomicron we had the B48, here we have the B100, but still the ApoC2 is not there, so still we have the nascent VLDL, or in other words it is the inactive, it is the inactive VLDL which is there. Alright, so liver will synthesize the triacylglycerol, package it in the form of VLDL and release in the circulation, where we get the nascent or the inactive VLDL. Same thing will happen just like chylomicron. It will go in the circulation, receive the ApoC2 and ApoE from HDL and become the active VLDL. The active VLDL will go to the extrahepatic tissue where the ApoC2 will activate the lipoprotein lipase. The activated lipoprotein lipase will now again act on the triacylglycerol and break the triacylglycerol into glycerol and the free fatty acid. The free fatty acid is quickly taken up by the extrahepatic tissue. Just like in chylomicron, the ApoC2 activates lipoprotein lipase breaks down the triacylglycerol, free fatty acid taken up by the tissue. This triacylglycerol is coming from the liver, so it is the endogenous triacylglycerol. This is the endogenous triacylglycerol, whereas in the chylomicron we have the exogenous triacylglycerol. So again the size will become smaller, all proteins cannot be maintained on the surface. The ApoC2 is returned to the HDL. So we are left with B100 and ApoE, comparable amount of triacylglycerol and the cholesterol. This is the VLDL remnant known as the IDL, which can be taken up by the LDL receptor and LDL receptor related protein, because we have both B100 and ApoE. Sometimes what happens, the delivery of triacylglycerol is nearly complete, so the size will become very small. It becomes so small that it is able to support a single protein. A single protein is there which is the B100. The size is much smaller, LDL. Most of the triacylglycerol is gone, so we are left only with the cholesterol. So this is the LDL particle. In the LDL particle, inside almost nothing is there. It is a very small particle. Whatever is there on the surface, the cholesterol along with the B100. Now because this particle is very small, it can be taken up by the LDL receptor in the extrahepatic tissue. Remember I told you extrahepatic tissue contains the LDL receptor, but it cannot take up the chylomicron remnant because the remnant is quite large. But this LDL is very small. It has released all the triacylglycerol content, so it can be taken up by the extra hepatic tissue in addition to the liver. Obviously the liver can still take up the LDL, but majority of it is taken up by the extra hepatic tissue. In this way, LDL is responsible for the delivery of cholesterol to the extra hepatic tissue. Remember, the chylomicron will transport exogenous triacylglycerol. VLDL will transport endogenous triacylglycerol. LDL will transport endogenous cholesterol synthesized by the liver to the extrahepatic tissue. All right. So this is the chylomicron and the VLDL. Let's move on to the HDL. Now we are going to be talking about the transport of lipid by HDL. So let's see. Okay. So some important points. Remember we had said the HDL is synthesized by liver. HDL is synthesized by liver as well as the small intestine. It is synthesized by liver as well as the small intestine. Both are responsible for synthesis. You can see for the HDL. HDL when it is synthesized is empty. There is nothing inside the HDL, and that is why it has the discoidal appearance. Discoidal HDL, you can see the structure is there and there is nothing in between. It appears as the phospholipid bilayer. There is nothing inside, so it appears as a discoidal structure, and this is the discoidal HDL. Mainly it contains the phospholipid and the cholesterol. The triacylglycerol is not there here, along with the various apolipoproteins, most important being the ApoA1. In addition, the HDL also contains the LCAT. Remember we talked about the function of LCAT. What does LCAT do? It converts the cholesterol into the cholesterol ester. So on synthesis we get the discoidal HDL. It has no content, it is flat. It is then released towards the circulation. By the time this discoidal HDL reaches the circulation, the LCAT has got the time to act on the cholesterol. Alright, what it will do? It will take up the fatty acid from phospholipid and add to the cholesterol, converting the cholesterol to cholesterol ester. That is the function of the lecithin cholesterol acetyltransferase. So the cholesterol will become the cholesterol ester. Remember the difference in property of cholesterol and cholesterol ester. We had said cholesterol is amphipathic, it is in the outer layer. Cholesterol ester is non-polar, it will go inside the core. So suddenly this cholesterol ester will start going inside. It will start making a structure. So you can think of it like a balloon. Currently there is nothing inside, but when you start blowing into it, it will become spherical. So as the cholesterol is converted to cholesterol ester, the discoidal HDL acquires a spherical shape. This spherical HDL is the HDL3. So by the time the discoidal HDL reaches the circulation, it gets converted to HDL3. This conversion is completed exclusively by LCAT. There is no entry of fresh cholesterol. Whatever cholesterol was there on the surface, that cholesterol only has been converted into the cholesterol ester by the action of LCAT, and this cholesterol ester, because it is non-polar, it will form the core and the spherical structure will develop. Alright, the spherical structure will develop and we get what is known as the HDL3. Now the HDL3 goes into the circulation and it receives the cholesterol into two primary ways. What are those two primary ways? First, most of the cholesterol which was there on the surface has been driven inside in the form of the cholesterol esters. On the surface there is very little cholesterol. When it goes to the tissues, there is a facilitative transport. I'm sure you remember facilitative transporter. It will transport along the concentration gradient from high concentration to low concentration. At this moment HDL3 has very low concentration of cholesterol, so the gradient is in favor towards HDL3. So when it goes to the tissue, SRB1 will transport the cholesterol from the extravascular tissue to the HDL3. As this cholesterol keeps coming, it is converted by LCAT into the cholesterol ester. So via SRB1, which is the facilitative transporter, the cholesterol is given to HDL3. In addition, ABCA1 and ABCG1, these are active transporters. ABCA1 and ABCG1, they are active transporter. Same for ABCG1. They are active transporter. They will transport the cholesterol in a particular direction. What is that direction? They transport the cholesterol towards ApoA1. The ABCA1 and ABCG1 will transport cholesterol towards ApoA1 in an active manner. So whenever the HDL3 goes to the tissue, if ABCA1 and G1 are there, they will identify the ApoA1 and they will give some cholesterol to HDL3. So what is happening? From the discordal HDL, first we got the HDL3. Now HDL3 is receiving cholesterol which is getting converted to cholesterol ester, so the content of the core is increasing. It will keep becoming bigger and bigger in size. After some time, it forms the HDL2. It forms the HDL2, which is a much bigger structure. Now this HDL2, which has large amount of cholesterol ester and also some phospholipid. From where the phospholipid come? Remember, cholesterol ester transfer protein will transfer the triglycerol, some amount of TAG. It also has some amount of TAG which is not shown in the diagram from the VLDL. So it goes to the liver. In the liver, the hepatic lipase and the endothelial lipase will act on the triglycerol. So the content will decrease, the size will become smaller. At the same time, the SRB1 will now start shifting the cholesterol from the HDL2 to the liver, because now the HDL2 has large amount of cholesterol. Because of these two reasons, what happens? The size of HDL2 becomes smaller and it gets converted back to HDL3. This is the content. It has large amount of the triglycerol also and the cholesterol and cholesterol ester also. So you give the triglycerol becomes smaller. You give the cholesterol also, it becomes much smaller. So it is reverted back to HDL3, goes back to circulation, and it can bring more cholesterol. There is one more fate which is not shown here. On the liver, we have what is called the HDL receptor. Remember, the HDL receptor is able to identify the ApoA1. So when HDL2 reaches the liver in the hepatic sinusoids, if the HDL receptor is there, it will identify the HDL2 via the ApoA1 and it will engulf the whole of HDL2, where it will be recycled. All the components will be recycled and they will be reutilized for synthesis of the new VLDL. So this is the transport of the cholesterol from tissues to the liver via HDL. Initially we get the empty or the discordal HDL. By the time it reaches the circulation, becomes HDL3. HDL3 in circulation receives cholesterol via SRB1, ABCA1 and ABCG1 and gets converted into the HDL2. HDL2, when it reaches the liver, has two possible fates. Either HDL2 is converted back to HDL3 by the action of the lipases and SRB1, or it is engulfed by the hepatocyte via the HDL receptor, where all the components and contents are recycled. One extra point to note: during the conversion of HDL2 back to HDL3, sometimes ApoA1 may get dislodged. This ApoA1 is either degraded by the kidney, or it can associate with some phospholipid and cholesterol, forming the pre-beta HDL. Pre-beta HDL is very, very important. Why? Because it is the most anti-atherogenic particle in the body. It is the most anti-atherogenic particle because its ability to take up cholesterol is very high, so it is able to extract the cholesterol from the plaques which are forming in the vessels, and therefore it is the anti-atherogenic particle. It is the most anti-atherogenic particle. It is formed during the recycling of HDL2 back to HDL3. It is dislodged from the surface of HDL2, and it can form the pre-beta HDL. Gradually, this pre-beta HDL will get converted to the discoidal HDL. So this is the reverse cholesterol transport by HDL. It will bring the cholesterol from the extrahepatic tissue back to the liver. So first we talk about the chylomicron, responsible for the exogenous triglycerol transport. Then the VLDL, which is responsible for the endogenous triglycerol transport. LDL responsible for the cholesterol transport to the extrahepatic tissue. HDL is responsible for the reverse cholesterol transport. It will bring the cholesterol from extrahepatic tissue back to the liver. Very briefly, I'll highlight the role of lipoprotein lipase. I already told you the lipoprotein lipase is anchored to the endothelium of capillaries, and it is acting on the triglycerol in the lipoprotein. Now the important point is, in which tissues will you find the lipoprotein lipase? In other words, which tissues will use the triglycerol in the lipoproteins? If the lipoprotein lipase is not there, the tissues cannot make use of the triglycerol which is there in the lipoprotein. So these tissues which make use of the triglycerol are the heart. It prefers to use the free fatty acid. Adipose tissue, it will take up the free fatty acid for storage. The spleen for various components. The lung for making the various lipoproteins like the surfactant. The renal medulla again for making the various molecules. Aorta again as a fuel. Diaphragm as a fuel. Lactating mammary gland for converting into the lipid for the milk. So these are some of the tissues which make use of the triglycerol in the lipoprotein, whether it is coming from the VLDL or it is coming from the chylomicron. Remember, the lipoprotein lipase is activated by ApoC2. If ApoC2 is not there, then the lipoprotein lipase will not work. So this is in brief about the lipoprotein and how they are transporting the lipids in the body. One thing which you will read further in medicine is the different hyperlipoproteinemias. From a biochemical point of view, I'll highlight three of them. The hyperlipoproteinemias are numbered nowadays: type 1, 2, 3, 4 and 5. Previously they used to have their names, but we don't use the names any longer, we use the number. Type 1, where there is lipoprotein lipase deficiency. The lipoprotein lipase deficiency can be because of various reasons. Either the gene is defective, or the translocation is defective, or the synthesis is not occurring. Okay, so the lipoprotein lipase deficiency, when it is there, see, the term used is deficiency. It is not absent but decreased in amount. So what happens when we consume the food? The chylomicron are formed. The chylomicron have to be quickly converted to the chylomicron remnant by the action of lipoprotein lipase. But because the lipoprotein lipase is deficient, this conversion is very slow. In other words, the triglycerol coming from the diet is used up slowly. Because it is used up slowly, the chylomicron will persist for a longer time, and primarily the content in this case will be the triacylglycerol. So when lipoprotein lipase deficiency is there, the chylomicron will persist, primary content being the triacylglycerol. Coming to type 2A, which has the strongest association with the atherosclerosis, here what we have the LDL receptor deficiency. LDL receptor deficiency, or sometimes we can even have the defective B100. Both are found recently. It has been seen that sometimes there can be defect in the B100 only. If B100 is defective, it will not be able to interact with the LDL receptor. So what will happen? The B100 is primarily used by LDL for uptake, because in the chylomicron remnant, in the VLDL remnant, in the HDL, we have the different ligands. In the remnants, we have the ApoE. For HDL, we have the ApoE1. So only thing which is left, which cannot be taken up, is the LDL. So LDL will persist in circulation, and primary content in LDL, it is the cholesterol. Last important from biochemical aspect is type 2B. In type 2B, we have the combined hyperlipidemia, means LDL and VLDL both are elevated, means the cholesterol is also elevated and the triglycerol is also elevated. Alright, so both triglycerol and cholesterol are elevated. In addition, we have the types 3, 4 and 5, the bis beta lipoproteinemia, the hypertriglyceridemia and the mixed hyperlipidemia, where respectively the VLDL remnant, the VLDL and VLDL and chylomicron all are elevated. Please remember the biochemical defects in these three, very important. They can come as a separate short note, so you should know about these, particularly for the type 2A. Type 2A is commonly asked separately as a short note, so you should be able to write this short note about the hyperlipoproteinemias. So that covers the various points that we wanted to talk about the lipoproteins. So today we have completed the discussion on the lipids. We talked about the ketone bodies, we talked about the bile salts, and we talked about the lipoproteins, how they are very important for the transport of the lipids. I'll repeat, this table that we drawn is very important. Alright, very important for your exam. Very easily you can get the question from this. When you are getting the question lipoprotein, you can quickly draw this table along with the brief description of the composition of the lipoprotein. If it is a 2.5 mark question, then you have to write this definition, the composition and the names. If it is a 5 mark question, then you have to mention about separation and the table also. So depending on marks, if it is a 2.5 mark question, just the structure of the lipoprotein and the different types of lipoprotein. If it is a 5 mark question, then structure of the lipoprotein, separation of the lipoprotein, and this table that we have drawn. Alright. So that is all for today. I'm closing the session here today. In the next session, we'll be starting with the amino acids. In the meantime, if you have any questions or doubts, you are most welcome to send them to me either in the app or over to me. So next class we will be having amino acids. Most probably it will be on Thursday. We'll confirm you via the app. Most likely it will be on Thursday. The flowcharts that we discussed have been taken directly from the textbook Harper, so you can have a look at that flowchart. But I think we discussed the flow very smoothly, and even if you don't draw the chart and you're able to draw the table that we've discussed, that should help you out with the lipoproteins. Any other questions? Alright, thank you very much. Take care, stay safe as discussed last time, and see you on Thursday. Bye bye.